Hi, good afternoon, everyone, and welcome to the people who are joining us online and also to the many people who are joining us in this room. It is very nice to see you all, and I really love coming to these talks and getting a couple of minutes before and after to catch up with people and just see them. But we are here today. This talk is being recorded. And the questions after the talk will be recorded. Although I assume someone told Ran something about how open it's being released. I don't know.
No, but
Okay. Well, we'll find that out. Okay.
well, probably, I signed a bunch of documents.
Yeah, we all sign a bunch of documents.
Probably in there.
But today it's a real pleasure to welcome Ran Blekhman, who is joining us today from the University of Chicago, where he is in the genetics department at the medical school and maybe is just an associate professor in the Department of Medicine. The reason that we really were excited about bringing Ran here is that I think one of the quintessential questions of genomics is how to combine data from large data sets. And in particular, one of the ones that we've been, ourselves very interested in and struggling with is when you have two data sets that are based on different indexes. So if one is the human genome, one is a microbiome, one is a this, one is a that. And Ran has really been a leader in helping to find ways to reduce complexity without losing all of the important signal there. So Ran was a PhD student at University of Chicago, unfortunately after my time, but we both got to enjoy those fabulous Chicago winters. And then went, and while he is now at the University of Chicago, he did do a circuitous trip around to other cold places in the US to live, including living in upstate New York, where he did his postdoc at Cornell with Andy Clark, and also then decided that none of those places were cold enough and he really needed to go start his assistant professorship at University of Minnesota in Twin Cities. Which is another great city I have visited. And so with that, Ran has gotten a lot of awards and fellowships, including being a Scialog Fellow from the Peter Allen Foundation, a Sloan Fellow. And really his work that he's going to talk about today, that is his funded work, is both on analyzing data sets that he's involved in generating, but also curation and processing of human microbiome data sets, a real value to the community. So thanks, Ran, and welcome to NHGRI.
Thank you. Thank you, Julie, for the very kind introduction, and thank you, everybody, for joining us. So, yes, exactly like Julie said, I'm going to talk a little bit about the genomics of host microbiome interactions. And I'm sure everyone knows what the human microbiome is, but just as a very brief introduction, there are trillions of microorganisms in and on our body. Some estimates say 10 times more cells than actually human cells in our body. There are thousands of different bacterial species, millions of different bacterial genes, which for me is really interesting as someone coming from a human genetics background. We have only 20,000 genes in our genome, so thinking about kind of the functional potential of all these microbial genes that are also on our body. They're found in several different body sites. I'm going to focus on the GI tract today mostly. And a favorite tidbit of mine is that the weight of these microbes combined is about 2.5 pounds, which is something I keep in mind whenever I step on a scale. And why is the microbiome important? Lots of studies have shown the microbiome is involved in metabolism. It can metabolize all kinds of compounds that we cannot, provide all kinds of nutrients that are important for us. It can interact with our immune system, and it can train our immune system, provide protection against infection, and it's also associated with lots of different diseases. And this is a slide. This is a figure from a by now a relatively old review paper, but just I like it because it kind of shows all the different body sites and different diseases. Lots of different diseases that have been associated with the microbiome. Probably by now, any disease you can think about, some studies have found an association with that disease in the microbiome. But just to highlight things, like obesity, metabolic syndrome, inflammatory bowel disease, colon cancer, which I'll mention later too. Lots of diseases, complex diseases, are associated with the microbiome. And for me, again, coming from a human genetics background, this is really interesting because, as you all know, this is a slide from the NHGRI website, from the GWAS catalog. There's been a lot of effort in the last 20, 30 years in identifying the human genetic basis of a lot of diseases and phenotypes. And this is, by now, this is a pretty old slide. I think now if you go to the website and you look at the same plot, you would almost not see the chromosomes because there are just so many loci that are associated with disease. But this just shows basically all the loci associated with human disease, all the loci in the human genome that are associated with different diseases. And I think I just want to point your attention to our things that are listed here, the names of these diseases, or the categories of these diseases, things like digestive system disease, cardiovascular disease, metabolic disease, immune system disease. So basically, all these diseases are also associated with the microbiome, but also have a human genetic basis to all these diseases. So basically, we know that host genetic variation, so human genetic variation, is responsible for a lot of the phenotypes and diseases we're seeing. And we know there's some kind of a link to the microbiome. The microbiome might be causal in some of these cases. It might be just correlational, but we know there's a link between the microbiome and complex phenotypes of disease. What I'm saying is if we want to understand the basis of complex host traits, we need to consider both host genetics and the microbiome. And the first step is basically to characterize the interaction between host genetics and the microbiome. Understand what microbes might be controlled by human genes, understand what human genes might control the microbiome. Can the microbiome affect host genetics by maybe adjusting or modulating gene expression in some host cells? This is basically kind of the big question that's driving a lot of the research in my lab. So having said that, this is what I'm going to talk to you about today. So I'll start by talking about host genetic variation effects on the microbiome. And I'll divide this part into three that would cover most of my talk. So I'll start with like a background. It'll be maybe for about 10 minutes or so, just kind of the background of the field, of how people are thinking in general about associating host genetic variation and microbiome studies. Some studies looking at common disease, or common genetic variants, some studies looking at disease-causing genetic variation in cancer. And then I'll talk about two bigger studies that we had recently. One talking about the heritability of the microbiome, meaning we try to quantify the genetic effect on variation in the microbiome. And we actually did this in a wild primate system. So I'll spend about maybe 15 minutes talking about this study. And then maybe about 15 minutes talking about a more recent study that is still unpublished, where we looked at global variation. So variation in the microbiome around the world using a very large data set that we collected from publicly available data of basically global variation in microbiome composition. And then if I have time, maybe in the last 5 minutes, 10 minutes of my talk, I'll talk about how then we're taking this microbial variation that we're detecting and trying to understand how it's affected the host. So what is the effect of this microbiome on the host? And we think one way the microbiome can affect the host is through changing gene regulation in host cells that interact with the microbiome. And I'll tell you about two ways we're kind of thinking about addressing this. One, by integrating data from different diseases and basically correlating data of host gene expression and microbiome data at different contexts and different diseases. But also, I'll kind of maybe mention a new approach that we're now establishing in the lab, where we're trying to use cell culture model to assess the effect of microbiomes from different human populations on host gene expression. Okay, so starting with a little bit of background, this, as far as I know, this is the first paper that actually looked at the effect of genetics, host genetics, on the microbiome. It was published a while ago now, like 23 years ago. And as you can see, there's a correlation here basically between genetic relatedness of the host and similarity in the microbiome. So it didn't actually have genetic data, but it just looked at relatedness of different individuals. And there is a correlation that is maybe significant. I don't know. But then newer studies used bigger and bigger sample sizes and found that there's actually some kind of a signal of host genetic potentially affecting the microbiome. This study by Turnbaugh et al. Actually looked at dizygotic and monozygotic twins and found that monozygotic twins who have much more similar genomes basically have more similar microbiomes than the dizygotic twins, although the difference is actually not significant. But they did find that individuals from that are unrelated have much less similar microbiome than individuals that are related or in the same family. So some signatures that might indicate there's a genetic effect on the microbiome. This is a study that we collaborated on with Seth Bordenstein's lab, where we found that there's actually strong effect of host ethnicity. So there's a self-reported ethnicity on the microbiome. We were actually able to use a machine learning approach to predict host ethnicity just for microbiome data, and relatively accurately. And this is a much more recent study from Ruth Lay's lab, where Ruth actually, what they did here is correlate microbiome variation from multiple different human populations and host genetic data. And they used this really interesting approach where you actually kind of correlate the tree, the phylogenetic tree of a microbe with the phylogenetic tree of the host. And you can find some cases, some microbes, where you can actually see a pretty significant correlation, meaning there might be kind of a case of a co-diversification or co-speciation of these microbes with their human host. Another previous study has actually tried to identify the genetic loci that are affecting microbiome composition. This is a study that was done in mouse using QTL mapping from the Benson lab, and what they did here is actually used an analysis where they can actually identify regions, loci in the mouse genome that are associated with the composition of the microbiome. And you can see here on the outside, this is basically the mouse genome, and each of these colored regions is a region, the locus in the mouse genome that's associated with certain taxon in this microbiome phylogenetic tree that's shown here in the middle. So a lot of indications there might be some kind of a host genetic effect on the microbiome. And all these studies, this was basically maybe 10 years ago, raised the idea of maybe we can actually use the microbiome as a quantitative trait and use genome-wide association approaches to identify genetic loci, genetic SNPs in the host genome that are associated with the microbiome. This is actually relatively straightforward if you know how genome-wide association studies work. You have the genetics of the host, but then you also have microbiome data from the same individuals, and then you define quantitative traits. So these are basically could be the abundance of certain taxa, it could be the abundance of pathways, the abundance of genes, it could be just global diversity of the entire community. So kind of measures of global information about the microbial community. It could be metabolic pathways. Some studies you can actually look at microbial SNPs, so genetic variation within the microbiome. And once you define your quantitative trait, you can just use a standard genome-wide association approach where you identify SNPs in the human genome that are associated with the composition of the microbiome. And this was basically what we did. This is I think the first paper that came out of my lab, where we basically used this approach with the Human Microbiome Project data to identify SNPs in the human genome that are shown here on the left that are associated with the composition of the microbiome, using the abundance, basically, of taxa as our quantitative trait. And you can see the taxa that were identified here. There's actually, this is the Human Microbiome Project data, so there's actually multiple different body sites with lots of different interesting results that we found here. I want to focus on one result that I think is the most interesting. Is also the result that was most validated the most times and has been since then in multiple studies that he's shown that this correlation is found. So again, this is the result of a genome-wide association study. In this case, what we're looking at is the abundance of bifidobacterium in the gut. And you can see here, this is basically the region in chromosome 2 in the human genome that's associated, where we found the association with the abundance of bifidobacterium. And what you can see is that this region overlaps the LCT gene. And this is interesting because the LCT gene is the gene that encodes the lactase enzyme, which is the enzyme that basically processes lactose in our body. And bifidobacterium is a microbe that's actually known to really like lactose. And some studies show that it prefers lactose over glucose. So this is a really interesting association, and I think this is a really nice figure that illustrates what I think is the mechanism. So LCT basically controls whether an individual's lactase persistent or not, right? If you have an active lactase enzyme, you can process the lactose that you get from your diet. And in those cases, if you're lactose persistent, you basically don't have any lactose available for the bifidobacterium to grow. And then you have low abundance of bifidobacterium regardless of your diet, right? But if an individual is lactase non-persistent and doesn't have an active lactase enzyme and still consumes dairy products, still has lactose available, then bifidobacterium would grow, and then you have a higher abundance of bifidobacterium in your gut. So this is a really nice kind of a mechanism by which genetics basically affects a metabolic process in your body, and maybe even affects dietary choice, right? That then in turn determines the abundance of a microbe in the gut. Another analysis you can do with host genetic information and the microbiome is not looking at common genetic variation, but specifically looking at disease-causing genetic variation, in this case in cancer. So this is a project of Michael Burns, who was the first postdoc in my lab and now is a already tenured professor. What Michael did here is actually look at tumor mutations using colon cancer tumors, and basically figure out for each tumor whether there's a loss of function mutation in a bunch of different pathways, and then find correlations, try to look at correlations, whether between those loss of function mutations in the tumor and the microbiome that's found in the microenvironment. So this is looking at the microbes that are actually attached to the tumor. And what Michael found here is that there's actually a pretty strong correlation between the existence of loss of function mutation in several pathways and the composition of the microbiome in the tumors. And again, it was able to use its kind of prediction approach to, just from the microbiome data that's attached to the tumor, predict whether there is going to be a loss of function mutation in certain pathways in the tumor. And as you can see, some of these pathways are really important for cancer, like MAPK signaling, canonical Wnt signaling, and so on. So a correlation, some kind of association of genetics with the microbiome, looking at our genome, but also looking at tumors, we're finding those kind of correlations in multiple places. And as a side note, like Julie said, we're also always trying to develop methods that might be helpful for people who also want to do similar kind of integrative analysis. And this is the work of Josh Lynch, who was a former grad student in the lab, and developed this approach called Hominid to integrate host genetic variation information and microbiome composition using a machine learning approach called lasso regression. And in this case, it basically gives you, when you input host genetic variation information in microbiome data, you get information on host SNPs that are associated with the composition of the microbiome, and then it uses stability selection to actually identify the specific taxa in the microbiome that are driving the association for each of those SNPs. That's available on our GitHub. Okay, that was kind of a little bit of a background about genetic variation in the microbiome, what we know previously. Now let me tell you about the heritability of the microbiome. So just to make sure we're all on the same page, when I say heritability, what I mean is from the population genetics perspective. So heritability is a statistic that basically estimates the degree of variation in a phenotypic trait in a population that's due to genetic variation between individuals in the same population. So basically, how much of the variation that you see in a trait is due to genetics as opposed to non-genetic factors. So it's a ratio, it's always between 0 and 1. If the heritability is called H square. If it's 1, it means the trait is completely genetic and there's no environmental effect. If it's 0, it means it's completely environmental and there's no genetic effect. People have been doing heritability analysis of many, many traits. Any kind of thing you can quantify, you can look at the heritability of that. And you can see, this is from a review paper, look at the heritability of a bunch of different various quantitative traits. You can see things like height, for example, always have very high heritability. In this case, the estimate is about 80% heritability, meaning it's highly genetic. There's still an environmental effect, but it's mostly genetically controlled. And you can see all these traits, kind of the heritability goes down until you get to things like personality traits that have much lower heritability, right? And if you look at the UK Biobank data, the UK Biobank is a very large data set where you have human genetic data. I think they have now 1 million individuals or so where they have genetic information. They also have a lot of phenotypes, a lot of quantitative trait information in that database. You can calculate the heritability of thousands of different traits, and then you plot the distribution of all of the heritability values. You can see this kind of distribution where very few quantitative traits have high heritability. So you have things like height, or eye color, things like that have very high heritability, those are here. And then most of the traits you can think about have heritabilities that are like somewhere between 0 and 0.2 or 0.3 or so. Okay, so what we want to do, again, look at the heritability of microbiome traits. Why do we want to do that? Well, first of all, it gives us a number to help us to quantify what is the impact of genetics versus environment on the microbiome, which I think is a really important question right now in our field. Also, any microbiome traits that are heritable are visible to natural selection acting on the host genome. So if we know something is heritable, only then we can say something about coevolution, which is also a really kind of important aspect right now in our field, trying to understand coevolution between the host and the microbiome. So looking at heritability, I think, is kind of a first step in order to do that. Another important note to make is that heritability doesn't really tell us anything about transmission. So sometimes you can think about heritability, especially if you're coming from the microbiome field, and think this means there's some kind of a transmission between mother to or father to offspring, right? This is not what I'm talking about. So when I say heritability, I only mean there's some kind of genetic control of this microbe. The way this microbe arrives or acquired could be maybe through transmission from the parents, but it could be from the environment. There's nothing that really indicates transmission when I talk about heritability. Okay, so I wasn't the first one to think about the heritability of the microbiome. This is a study, the first study to actually look at the heritability of the microbiome. This is from Ruth Leigh's lab, where they used twins data again. And the idea is what you can do when you have monozygotic and dizygotic twins, you can actually look at the similarity in abundance of taxa in the microbiome in monozygotic twins and dizygotic twins. And if you have higher similarity in the monozygotic twins, that could be an indication that this microbe can be genetically controlled. So you can actually use this information to quantify the heritability of the entire microbial phylogenetic tree, and you can see that here on the left-hand side. And as you can see, most microbes don't have very high heritability. It's actually very close to zero. But in some cases, you can see microbes in this tree that actually have pretty high heritability, and sometimes it's even more than 0.4. Some of these taxa, Christensen and Lechier, shown here, was kind of pinpointed as the highest, the most heritable gut microbial taxa in this study. And this is a review paper from 2017, I think, that kind of basically synthesized information, results from several different microbiome heritability studies. And what they found is there is some consistency. So some microbial taxa appear again and again as heritable. One of them is Christensen and Lechier, which you can see somewhere here. But on the other hand, there's also a lot of taxa that are found to be heritable in only one study and not the other. And this is kind of surprising. And also another thing that's pretty surprising is that some of these studies actually find really high heritabilities for some taxa. They get to like 0.5 or even 0.6, which is kind of surprising to me as a human geneticist. You see this high heritability for this kind of trait. And this is an older study. This is a newer kind of synthesis from a review paper that we're finishing up that actually shows, it's a little hard to see, but it basically takes the four largest microbiome heritability studies. Since then, there were a lot of, this kind of shows all the microbiome heritability studies, how many, like the sample size that we see. And you can see that in the last few years, there were a bunch of studies that are much bigger and get to even close to 10,000 individuals by now, right? So taking these four largest studies and doing kind of similar presentation where we're looking at the microbes that are found to be heritable, we see a similar pattern where a lot of these taxa are found to be heritable in one study and not the others, right? But what you can see is that we don't really see that really high heritability anymore. So now it's like 0.5 is probably the highest, and most taxa are much well below that. Right? So patterns are changing a little bit. And some of the challenges when you think about the heritability of the microbiome and how to study the heritability of the microbiome is the microbiome is influenced by so many environmental effects that it's really hard to get a really good quantification of heritability. This is an example from a study from the Zernakova lab where they basically had a cohort of maybe 1,000 individuals where they actually quantified all their dietary components, all their medication at a very high detail. And what they found here is that almost any dietary component you can look at is correlated with some kind of difference in the microbiome. So lots of different things like how many fruit does someone eat, how much pastry, how much yogurt, it's all correlated with differences in the microbiome, but also obviously things like medication and so on. So lots of really big environmental effects, and it's really actually hard to quantify those environmental effects, right? If you know some of the human studies that try to look at the microbiome and try to quantify that, usually you have like very big questionnaires asking people, so what was your diet over the last year? How much bread do you eat per week? Stuff like that. It's really hard for people to know. So sometimes they don't fill it out. Sometimes they fill it out just not accurately. So it's really hard to get a good estimate of these. And also something that's really important for the microbiome, it's actually really dramatically variable day to day. So the microbiome changes almost every day, and you have those changes. It's really hard to quantify because to do that, you need multiple samples, lots of longitudinal samples, which is really hard to get if any of you have done any human studies. Most of the human heritability studies that I mentioned have maybe one, maybe two samples, I think, tops. I haven't seen any study that had more than two samples. So, because it's really hard to coordinate those kind of sample collection. Okay, so what do we do? How do we resolve this? Well, we don't look at humans. Instead, we're looking at wild primates. This is a study that was a really close collaboration that we have with the Amboseli Baboon Research Project. So this is a longitudinal research project that has been using wild baboons in the Amboseli in Kenya as a model to study lots of different traits and phenotypes, including behavior, ecology, genetics, and aging. It was founded more than 50 years ago by Gene Altman at Princeton. And since then, there's been basically near daily observations of animals in this population, including looking at what they eat, who they interact with, how they use their habitat, and the most important information that we have is kinship. So we don't have genetic data, but we have the entire pedigree of everyone in the population. So we have the genetic relatedness of each individual. If you think about the microbiome, look at heritability, it's very similar to what you do with the twins, where instead of having monozygotic and dizygotic twins, we actually have the genetic distance, the relatedness between any pair of individuals. So we can use a similar approach to estimate the heritability. And the important thing is they have fecal samples that have been collected and preserved since about the year 2000. So initially they were collected for doing hormonal analysis, but we decided we can go back to the freezers and take all these samples and sequence the microbiome, and then use all this rich metadata to actually get an accurate quantification of microbiome heritability. And there were a lot of people involved in this. This is a really big effort in going and collecting their thousands of fecal samples. I have to thank Jenny Tang and Beth Archie, who are co-directors of the Amboseli Baboon Research Project and are amazing collaborators. There are a lot of people who were involved, obviously, collecting all these samples over the years and processing them and processing the data and everything. I'm going to focus on just the heritability analysis that was done by Laura Greeneisen, who was a postdoc in my lab and now has her own lab. And you can see her here in this picture right after collecting a fecal sample from a baboon. Some reason she's not wearing gloves, I'm not sure why. There were other studies, so I'm just going to talk about the heritability analysis we did of the data. There are a lot of other studies led by Beth Archie that looked at changes in the microbiome over time, longitudinal changes within individual variations in the microbiome. Really cool research that I'm not going to mention today. I'm just going to focus on the heritability of the microbiome. This is what the data looks like. So we have in overall 16,000, more than 16,000 microbiome samples that we sequenced here. You can see the data here, or the samples here. So on the x-axis, you have the age, and on the y-axis, you have the individual. So each row is basically an individual, and all the samples taken from one individual. So as you can see, we have about a little less than 600 individuals, but lots of samples from each individual. For some individuals, we have dozens of samples, 28 samples is our median. And the samples basically cover, in most cases, cover the entire lifespan of an individual. So we can actually get a good handle of variation in the microbiome and day-to-day changes in the microbiome. We did 16S sequencing because that was the cheapest, easiest way to get that many samples sequenced. Now we actually just finished doing metagenomic sequencing on the same samples, and we're starting to dig into that and look at like heritability of functional profiles of the microbiome, look at transmission of microbial strains in this population. Some really exciting stuff that will be coming out in 5 years, I don't know. And the most important thing is that we have the detailed information on diet, relatedness, social interaction, environment, lots of really cool metadata. This is what the data looks like. I don't know if you've seen a stacked bar plot of microbiome data before. This is basically every column is one individual, or one sample, sorry, and you have 16,000 columns here, and each color basically represents one taxon. So you have the abundance of each taxon, each sample. There's a really big, so the take-home messages from here is it's a really big data set. The second is that there's a lot of variation in the data, which is really important. With no variation, there's no way you can quantify heritability, so variation is good. And I'll say sometimes you can use data to make art. And this is what the data looks at, the same data, but now it's ordered by the date of collection of each sample. So each column again is the sample, same thing, and you can see the dates where the samples were collected. The one thing you can note here is that there's kind of this cyclical pattern where in each year you have basically the microbiome diversity going up and down, with some taxa going up, some taxa going down. This is because of the seasons. There's a wet season and a dry season in this population, and I'll come back to that in a bit. Okay, so what Laura did here is basically define microbial phenotype, microbial quantitative trait in the same way that I mentioned before. Basically, she defined 7 community phenotypes. Those are like diversity of the community and things like that. Then she defined hundreds of taxon abundance phenotypes. This is just how much of each taxon you have. But then also binary phenotype, which is just presence absence. Is a taxon present or absent in the sample? So more than 1,000 different quantitative phenotypes. And then she basically ran a model, a very big model that included all the microbiome data, basically each of these traits, and assessed the heritability, controlling for all these covariates that we had for the data. So things like age, sex, social interaction network, like which individuals interact with each other, maternal effects, to know that it's not just an effect of the mother like transmitting, it's actually genetics. And obviously technical effects, anytime you have a very big genomic data, there's going to be some technical biases you have to correct for, so we corrected for all of that. Just that bullet point took like a year to figure out, but we did it. And then all the environmental effects that I told you about. So things like season, rainfall, diet. So just to kind of give you an idea, this is what the diet data looks like. Right, so this is not microbiome, this is the diet data. So basically for each time point, for each microbiome sample, we have a complete dietary composition of what was happening at that specific date. And we can look at the components of the diet. And you can see the same kind of effect of the season here even more clearly. So during the wet season, you have more diversity in the diet. During the dry season, there's much less dietary components, and a lot of it is composed of corms, which is this blue, kind of teal color. You can see that here. So we can actually include all these dietary components in our model. So we can actually control for this in our model. So Laura did all that, and what did she find? Well, she found that basically all of the microbiome traits, or essentially all of them, more than 95%, are significantly heritable. Any kind of trait that she looked at. So the relative abundance data, no matter which way she transformed the data or not, which way she analyzed the data, she took phylogenetics into account or not, the presence-absence traits, these were all significantly heritable. However, when you look at the distribution of heritability values, this is the x-axis showing this is the distribution of heritability values, you can see that they're all in this range of very close to 0 up to like 0.2 or a little bit more than 0.2. So heritability estimates are actually relatively low. And this is actually not surprising to me. Initially I was surprised, but then I was actually not surprised, because then I remember this is the distribution of heritability values from the UK Biobank data. And this is kind of what you would expect for almost any trait you look at, that there's this kind of distribution, and almost all traits are in this area where there's a genetic control, there's some kind of genetic effect, but the genetic effect is relatively minor. And especially with the microbiome, I think it makes sense. There is a genetic effect, but genetic effect is small compared to the environmental effect. And this is what we're finding here. If you look at the taxa that are most heritable, the most heritable quantitative trait is actually not a taxon, but the first PC, one of these community phenotypes, the first PC of the microbiome data, which you can see here. The taxa that are colored in red are the ones that were also found to be heritable in human studies. For example, Christensen and Lakeshia, you can see it's heritable in humans, but it's also one of the taxa at the top in our analysis, and that kind of prompted us to try to see if there's any correlation in the heritability estimates between human studies and our baboon study. And what we found is actually a correlation, a significant correlation between the heritability we're finding in the baboons and the heritability that was found in human studies. And I don't want to overinterpret this result, but this just might indicate there's some kind of common genetic control, maybe conserved genetic control across species. I also mentioned the 2 seasons. One thing we can do is actually split the data into 2 and analyze the heritability of the wet season samples and the dry season samples separately. And then we can look at the correlation between them. And what we're seeing here, that they're actually very highly correlated, but the correlation is shifted. such that the heritability is higher in the dry season. So for almost any trait you look at, the heritability is going to be higher in the dry season compared to the wet season, which makes sense because there's less, in the dry season, there's less dietary diversity. So there's actually more space for genetics to have an effect on those microbiome phenotypes. Another thing we wanted to assess is whether the fact that they had a big sample size and also longitudinal samples had an effect on our results. So first thing we did is just subsample from our sample just to see what the effect of smaller sample sizes. And what you can see is that when you have smaller sample sizes, you get this huge range of heritability estimates, which I think is probably just noise from having a very small sample size. And as the sample sizes grow, you can see it kind of converges to heritability estimates, what we're getting, which is like 0.2, 0.1 or so, which kind of falls in line the fact that these human studies that had smaller sample sizes also, in some cases, got pretty high heritability estimates for some taxa. We can also show here that if the larger the sample size, the more taxa you're detecting as heritable accurately. And we're seeing that once you get to like 10,000 samples, then you're getting to this like high accuracy prediction of the heritability. Another thing we wanted to do is the effect again of the longitudinal data. So what we did is we subsampled this number of samples we had per individual to 1, 2, 5, 10, or 20 samples per individual, and we plotted this along this line here. The purple dots are our data, the heritability, and the yellow dots are from human studies. And again, the human studies are in yellow. All of them had either 1 or 2 samples, but per individual. But what you can see is that the more samples per individual we have, the more the larger number of heritable taxa that we're finding. Again, indicating this longitudinal data is really important. And I think longitudinal data is actually, we're going to find, is going to be really important, not just for heritability analysis, but for any kind of microbiome analysis. Because of this kind of day-to-day variation, I think having longitudinal data is super important for any microbiome study. And just as a side note, we noticed there actually was no like good way to actually visualize like really large longitudinal data sets. So Laura developed this tool which is meant to just visualize longitudinal microbiome data using this horizon plot approach, and it's available on our GitHub here. Okay, so I told you a little bit about the heritability of the microbiome. Now I'll tell you about another study, a new study that we have that's supposed to come out pretty soon, looking at global variation in the microbiome, variation basically across the globe. And that's important because we had a few studies, so a lot of labs actually looked at kind of the importance of looking at the microbiome across different human populations. Just one example from my lab, this is Elise Morton, was a former postdoc in the lab, and what she found is there's actually a big difference in the microbiome. If you look at these microbial composition in distant human populations, including hunter-gatherers, farmers, and you can see some pretty big changes in the microbiome, including some specific taxa, like bifidobacterium, which I mentioned before, other taxa that are very different. There's another study from Andres Gomez, who was another former postdoc in the lab, in this case, also finding those kind of patterns, but also discovering this taxa where you had this gradient effect where hunter-gatherers has much, much higher abundances compared to farming population, then compared to the US, where some taxa are completely absent. And some examples are Prevotella, that shows this kind of gradient across populations. And it's really important. There were a few studies that have shown why this is important. There's an example from a study from the Sonnenberg lab that has shown that this kind of variation, those kind of taxa that are found in hunter-gatherer population, for example, are actually really important for healthy development. And they showed using kind of an animal model approach where it's actually really important to study those taxa and understand those differences. Now, the problem is that there's actually a really unequal representation of microbiome sampling basically in the field, right? You can see this figure here on the right-hand side. This is a study we published a couple of years ago where we actually looked at all the publicly available human microbiome data. We actually didn't look at the data itself, but just for each sample, we figured out where it's from. So what country was it sequenced in, right? And then we plotted this along basically over time and the number of samples. You can see there's a huge increase in how many microbiome samples have been sequenced over time. But you can also see this pink area here, which is samples from Europe and Northern America, is completely dominating the samples that are being sequenced. And you can see here on the right-hand side, this is actually the relative kind of human population. You can see that maybe about 75% or so of microbiome samples that we sequence are from Europe and Northern America, but only maybe 15% of the world population is from these regions. And so there's really not a lot of studies that actually looking at kind of human populations that are not from Europe or Northern America. And the reason is kind of makes sense, right? Because these studies are really small. It's really hard to coordinate. It's really hard to fund those kind of studies. So it makes sense. There are a lot of a bunch of kind of small studies. And it's really hard when the studies are really small to actually look at global patterns, like differences around the world in microbiome composition. And if this figure looks kind of familiar, we were really inspired by this figure that was published in, I think, 2019 by Martin et al. I should add the citation here, but it did a very similar analysis for human genome data. Right, and what they found here, a very similar pattern. Almost all human genomes are in sequence are from European ancestry, and this is again not representative of the actual global population. But this bias in human research and human genome research is actually being addressed by a lot of like top-down efforts to specifically go and sequence more genomes from underrepresented human populations, even previously by the Human Genome Diversity Panel, HapMap, 1,000 Genomes, all had a specific goal of sequencing more human populations, and now H3 Africa, the CZI, Ancestry Networks, all specific like funding opportunities to sequence these kind of world populations, but we don't actually have anything like that for the microbiome at the moment. So how do we solve this? What can we do? Well, there's a lot of data, petabytes of data that are available at the SRA. And the SRA is I don't know if you've never heard about it, but whenever you publish a paper, you have to submit your data. It's really annoying. You have to spend like 2 days figuring out how to submit it and upload it, and then it's there, and then the NIH has to spend a lot of money to maintain it and make sure it's like preserved forever. But not a lot of people are actually using all this data. But it's there, and people from other fields in genomics are actually using a lot of the SRA data to do this kind of integrative analysis. So there's an example of one study from our collaborators from Casey Green and Sean Davis's lab, where they used a similar, this kind of approach, looking at RNA-seq data. They integrated all the RNA-seq data that's publicly available at SRA, and they found some interesting biological results. And our question is, can we actually use the SRA data to characterize global variation in the microbiome? Can we take all the SRA data, all the microbiome data at SRA, and actually look at the variation in the microbiome around the world? And the reason we thought we could do it is because, specifically, if you look at 16S data, it is actually pretty small data sets, right? Each 16S data set has maybe 10,000 reads or so, and that's opposed to RNA-seq data where you have millions of reads, for example, right? So this is actually especially amenable to this kind of integrative analysis. So this is what we did. This is the work of Rich Abdul and Samantha Graham from my lab. We call this the Human Microbiome Compendium. The goal, like I said, is to integrate all publicly available human microbiome data, process it all into one pipeline, similar, identical pipeline, and basically at the end reach basically a database that has all the samples in the column, like one matrix where you have all the samples and then all the taxa. And you have the abundance of every taxa in every sample by this kind of unified processing pipeline. This is one of those cases where I show a slide and talk about it for 30 seconds, but this is like a year and a half of work. So Rich and Samantha always hate me when I don't talk more about the QC and all those things, but I think this is less interesting. It's important, but maybe less interesting. But I'll just say that we now process all the 16S data, specifically looking at gut microbiome as the first step. And the next step, we're going to expand it to all the other body sites and also look at metagenomic data as well. The end product, I would say right now, we have about a little less than 170,000 samples included in this human microbiome compendium. This is just some results. You can see, like I said, the read counts per sample. The median is about 40,000 reads per sample for 16S, although there are some samples that have like millions of reads in their 16S study. Not sure why. You can see this is a nice plot that might be important for later, but what you can see here is basically how many unique taxa we're detecting based on the number of samples. And as you can see, the more samples we have, we're detecting more taxa, but like at the order level, maybe after like 10,000 samples, 20,000, 50,000 samples, we're not really detecting anything new, right? But for at the genus level, we are detecting new taxa, although it is kind of plateauing pretty soon. And these are some of the taxa we're finding. They're pretty common. An interesting result here, there are some taxa that are actually found almost everywhere. So almost every microbiome around the world, you're going to find, bacilli or Clostridia, which is, I thought was pretty interesting. This is what the data looks like. Again, a stacked bar plot. In this case, it's 160,000 samples. Not a lot to learn from this, but again, there's a lot of variation in the microbiome around the world. These are the taxa. Again, we wanted to see what is the representation of different world regions from our microbiome, this global microbiome data. Again, we're seeing exactly the same pattern. We can also use this funny thing where you can kind of change the size of countries based on how many samples there are, which gives you this kind of a little funny, but also a little sad picture of with some world regions that completely disappear because there are almost no samples from these world regions. We can look at global variation in this microbiome data. I'm going to skip this just to make sure I have time to get to the most interesting result, which is this. I think is probably the take-home message from this analysis, which is doing basically the same kind of taxa detection approach, but now instead of looking at different phylogenetic levels, we're just looking at different world regions, right? And what you're seeing here is that in world regions like here in the pink, like Europe and Northern America, we're probably reaching a plateau, right? So we can sequence more samples, but we're not really detecting any more new taxa. But there are some world regions, especially some world regions where there's almost no sequencing done, and it's really even hard to see because there's so few samples. But for example, North Africa and West Asia, Latin America and the Caribbean, where we're not even close to getting all the taxonomic information we can get, or getting all the genetic diversity we can get from the microbiome. And we're thinking, is this just because of the higher coverage? It's not. And you can see here, based on coverage too, that we can sequence more samples from Europe and Northern America, but we would need many millions of reads to detect even one new taxon. While in other world regions, we're getting a lot more for less in terms of how much sequencing we want to do. We can also identify specific taxa that are different between world regions. This is just pairwise analysis, looking at how many taxa are different between each pair of these global world regions that we're looking at. And you can see there are a lot of differences. And basically any taxon we looked at is different between at least one pair of world regions. So a lot of differences between these world regions. This is just looking at which taxa are different between Europe and Northern America and the other world regions. And you can see, again, almost every taxon, aside from like just a few, is significantly different between Europe and Northern America and at least one other world region. So lots of differences in the microbiome. You can highlight some of them using this kind of plot. So for example, Bacteroides, you can see here, has this distribution where it's found in Europe and Northern America. In pink here, it's actually the higher abundance, but you focus here on the blue, it's much shifted. Similar thing, the opposite direction is Prevotella, as you can see here, which is much lower abundance, and we know it's actually absent in a lot of samples from Europe and Northern America, but it's much higher abundance in sample from this orange here, which is sub-Saharan Africa. So you can see it's like basically the opposite pattern for some taxa between Europe and Northern America and some of the other world regions. We also found a similar kind of association between microbiome variation and global composition based on not just this larger world region, but based on country. And we can see this clustering that's based on country, where the microbiome is from, is actually highly significant. And the final note, we want this to be like a really kind of a resource for the community. So we put all of this, made all this available online. If you go to microbiomap.org, you'll find all the data there. You can download the data in different formats. You can download a database that all the data. We have an R package where you can kind of look at specific microbes. And we have those recipes in R where you can actually give you some code snippets that you can use to, like, can I get all the microbes that are higher in this world region? Can I get the microbiomes coming from this world region? Can I get the things that are different between this and that? So you can actually kind of hopefully get started pretty quickly. And the website also has some nice visualization. You can pick certain countries, pick certain taxa, and see how their distribution changes around the globe. Okay, so I think I have maybe 5 more minutes or so. In the last 5 minutes, I'm going to kind of just touch on a couple of studies that are some of them are published recently, some of them are upcoming, where we're trying to understand what is the impact of this variation in the microbiome between human populations, between health and disease, on the host. What is the effect of the microbiome on host cells? And we think one way the microbiome can affect the host, one kind of mechanism that's pretty clear, that's really emerging recently, is that it's changing gene regulation, gene expression in the cells that interact with the microbiome. This has been shown in multiple study systems, including the zebrafish and the digestive tract, and the gut of sticklebacks, and in the mouse, and the intestine, and the skin. And every time, usually the studies have germ-free animals. When a microbiome is introduced, you see a big change in gene expression in the cells that interact with it, whether skin cells, intestinal cells, and so on. So we want to look at this effect using correlational analysis between the microbiome and host gene expression, and see how that changes between health and disease. And this is the work of Sambao Priya, with a former PhD student in the lab. And what Sambao did here is look at data sets where we have RNA-seq data from the host and microbiome data, all from biopsies, where we actually have RNA-seq data from the cells that interact with the microbiome and microbiome data from the same tissue samples. And she had 3 different cohorts, 3 different disease cohorts, including colon cancer, inflammatory bowel disease, and irritable bowel syndrome. And she developed this approach that basically uses machine learning techniques that are complementary, including sparse CCA, that gives you basically this kind of association between clusters of microbes and clusters of host genes, and gives you kind of associations at the kind of the pathway functional level. And at the same time also doing lasso regression, which I already mentioned, but gives you this kind of pairwise association between specific host genes and specific taxa. And the code, again, is available for this framework for integration. I'll just kind of mention the highlight, which when you look at the host genes that are associated with microbes across these different diseases, we're seeing some host genes that are similarly associated with different, are all associated with the microbiome in these 3 diseases, but they're associated with different microbes in each disease, maybe indicating different pathways. The most important result, I think, is if you look at the microbes that are associated with the host genes and clustered by the microbes, we're seeing basically the same microbes appear, and these are microbes that have been previously associated with all 3 diseases, including Staphylococcus and Streptococcus. But the microbes in each of these diseases, the microbes are associated with different host genes, which again might indicate you might have a different pathway for each microbe to kind of change physiology in each of these diseases, although we keep seeing those microbes come up in all those disease studies. And as a final note, this is another kind of study that we have that's still in the works, but we're developing this cell culture-based approach to actually assess the effect of the microbiome on host gene expression. This is based on colonic epithelial cells. And right now we're trying to move into organoids, colonic organoids as well, that we're treating them with microbiome, basically complete microbiome communities we extract from fecal samples. So we can take a microbiome sample, extract all the microbiome, or fecal sample, extract all the microbiome, based on the same approach as used for fecal microbiome transplant, to extract the microbiome from fecal samples. And we basically treat host cells, and then measure the response of host genes using RNA-seq or any kind of genomics approach. This approach has a lot of advantages. It's easy to do, high throughput. It's very controlled, so there's no environmental effects. It's a tightly controlled system. And we can use any kind of genomics techniques that we want. We use RNA-seq, we use ATAC-seq, whatever we want to basically assess the effect of these microbiomes on host cells. And we can look at it in different contexts. We're thinking about doing this in health and disease, so taking microbiomes from, for example, IBD patients and healthy individuals. One project that we have that we're now finalizing is actually looking at microbiomes from different human populations. There's a collaboration with the Global Microbiome Conservancy, which is a biobank of stool samples from different world populations. And we're using these stool samples with the same kind of experimental technique to basically measure responses of host cells to microbiomes from different populations or industrialized and non-industrialized hosts, basically. It is in collaboration with Francesca Luca and Sabrina Arif. And we're hopefully going to get some results from this pretty soon. But the idea, there's some very, very preliminary results. The idea is that we can actually measure host pathways that are downregulated only in response to the industrial microbiomes or the non-industrial microbiomes, and some initial results tell us that some specific inflammatory responses to the industrialized microbiomes and not to the non-industrialized microbiomes. And then we can also pinpoint, because this is RNA-seq data, we can pinpoint the specific host genes that are responding to each microbe. So basically correlated with the abundance, the response of that gene is correlated with the abundance of the microbe in the microbiome that we use to treat. So we can like pinpoint specific microbes that are directly affecting the expression of host genes using this approach. Okay, I'm going to end there. I'll just kind of summarize. I told you a little bit about the host genetic control of the microbiome. We can use standard genome-wide approaches to identify host genes that are associated with the microbiome. We can quantify the heritability of the microbiome. What we're finding that the microbiome is heritable, so a lot of the components of the microbiome are significantly heritable, but the heritability is not very high. And the environment is more important than host genetics. We also look at global variation. I showed there's huge variation in the microbiome around the globe, and we think that there are more efforts can be done to kind of select and sequence in a more representative way. And then I'll tell you kind of how we're thinking about trying to understand the effect of those variations in the microbiome on the host, specifically through changes in host gene regulation. And that's it. I want to thank everybody involved in these studies. I think I already mentioned everyone that was contributing to the specific project that I told you about. This is our funding, and thank you very much for your time. Happy to take questions.
Okay, thanks so much, Ran. And please, for the people online, you can put your questions into the Q&A box, and we are monitoring that. And we will start in the room with Nikki.
Can you hear me now? Okay.
Yeah.
So I want to commend you for the integration of the microbiome, because I think that is going to be a great resource for the community. And so I'm very excited to see that. I know that you're doing it also in a way under the globe of genetics in the grand of population genetic kind of thinking, but I wonder what other metadata is available for all of this.
Yeah.
So I am a clinician and immunologist. I care. Is there a possibility to mine under disease state or to mine under other things that might be of interest to many of us within your huge data set?
This is a really good question. Yeah. So this is one of the biggest challenges when looking at SRA data is that we focus. One of the reasons, not the reason, one of the reasons we focus on this kind of analysis is that the geographic location is one of the more
straightforward,
accurate metadata that there is available in SRA. I would tell you a lot of the other types of metadata is either not there or just not accurate, or it's just not reliable. So we're finding some metadata, so in a lot of cases it's not correct, and it's obvious from the data that this metadata is wrong. Sometimes, you have a sample, it says it's human microbiome, gut microbiome, but it's not human, it's not gut, and it's not microbiome, right? It's more often than you would think.
Yeah.
So what we're doing now to try to get this metadata to be reliable is we're collaborating with Arjun Krishnan, who's at the University of Colorado, together with Sean Davis, also at the University of Colorado. We're basically trying to use this kind of machine learning approaches that can read the papers, and from the paper, so each of these samples can be attached to a project. Each project could be attached to a paper, read the paper, and try to get basically automatically get the specific type of metadata, like disease information, or we've already used this with some metadata I haven't told you about, but to get information about, like technical information about the specific extraction kit that we would use for each microbiome, because that's relatively standardized. Again, this is not part of, it was not reliable part of the metadata on SRA, but when we went to the paper, this was actually something we could extract. And we got, yeah.
Thank you for doing that, because that's going to make it so much more useful in many ways. Now, on the host microbe interaction, that for me is even more interesting. In your paper where you're looking at tumor-specific responses, are the microbiota the ones that are normally there that you're importing, or are they on the patients that you're looking at the host response, and are they local on the tumor? Because from my end, also kind of the local, there's many things that all of us can think about, anywhere from secreted products in the blood that can go to a tumor, but also attachment onto a specific cell and availability within the region. So I wanted to kind of get a little bit more information of what is involved in this study, but also other ways that you're thinking about.
Yeah, for this the colon cancer study, the microbiome is all from the tumor samples that were extracted. So from the same tumor sample, we did whole exome sequencing to get the mutational information and microbiome data, like 16S, from the same sample. So this is supposed to be like the microbes are attached to the tumor. Yeah, it's kind of challenging because there's not a lot of microbial information there. There's not a lot of microbial material in those samples. So I think 16S could work, but it's still a little challenging. Yeah. Right. Mm-hmm. Yeah. I agree.
Thank you for your talk. I have two questions. I think one is pretty boring and one is more interesting. The first one is for your global data set meta-analysis. You started to talk about this. Is how much are you thinking about technical variation and like facility effects and batch effects and things like that?
That's a very good question. Yeah, we're always thinking about batch effects. Any kind of genomics lab should be always thinking about batch effects. In this case, for this study, for some of the analysis that, like for example, the differential abundance analysis that look at different texta, you could run like a model where you can include all the technical covariates you have access to, which is what we did here. Like anything we know about the samples, the time where those samples, year they were sequenced in, whatever, right? Some analysis is hard to control for that, right? So we included in that analysis, we included like the project, like the batch, basically in this analysis. And like I said, the answer to a previous question, we did get technical information about like how the samples were extracted, the extraction kit. We included that as well as the covariates. Any other batch effect? So my belief, I don't know, is that because the data set is so big with 160,000 samples, even if there are like batches, there are just so many small batches that none of these batches is going to have an actual big effect on the results we're seeing between. Yes.
Is the data set big enough basically for that?
That's kind of what I'm thinking. There's no way we can actually test that hypothesis. We know this is like true based on like other analysis people have done for RNA-seq and RNA-seq data, is that when you integrate like 500 different batches, then any kind of specific batch effect is just going to wash out compared to the actual difference that you're seeing between your conditions of interest. That's my hope, yes. But I don't know, still could be something, yeah.
Thank you. And then my other question is, so for the heritability studies, I think it's always interesting to think about how much of a trait is heritable versus environmentally influenced. But I think also what's really interesting is when genetic variation mediates an environmental effect, whether that's through behavior or something else like that. Yeah. And so I'm wondering, with something like the baboon study, do your statistical methods allow you to see whether there are any microbial traits that are subject to that kind of genotype by environment interaction?
Yeah, so that's a really good question where, you have a heritability and the heritability is different. So we don't have genotype data here, which is, the issue, but we can look at the heritability. And one thing we did, like, for example, the analysis of looking at the heritability in different seasons. And you can see it's correlated, but there are differences, right? There are some taxa that are more heritable, like all of them are more heritable in dry season, but they're not all completely along diagonal, right? Yeah, so there is an environmental effect on heritability, right? So something to keep in mind, the heritability is not like a set number, like this microbe is always gonna have this heritability. The heritability depends on context, it depends on population, it depends on a lot of things, right? And which is what we're seeing here, but this is expected, this is like what people know from heritability of, any other trait that changes, right? But I think one thing we can do we plan to do with our organoid approach, where we treat them with microbiome, we can actually look at SNPs in the host, and SNPs that kind of modulate the response to certain microbiomes. And I think that might be a way we can try to answer that question. Yeah.
Cool. Thank you.
Hi, Ren. Lovely talk. I wanted to ask about the ethnicity and machine learning prediction. So you said that the model could predict ethnicity based on the gut microbiome, but I'm wondering, like, how accurate it would be to say that it's entirely ethnicity.
Yeah. So, right. So this is self-reported ethnicity, and it's not. It doesn't mean that the differences in microbiome are due to ethnicity,
Right.
or it means it's just a correlation, right?
Mhm.
And it's not. Doesn't mean it's genetic. It could be different diets. It could be different, environmental effect. It could be a lot of things, right? So it's just like a statistical prediction. So I don't want to give it too much meaning aside from that. Yeah.
Okay. There are a couple questions online, and we're going to send them to you, or we'll answer them ourselves, and we'll just say that, because I think at this point we're sort of at 10 minutes after the hour. So I hope we can all thank Ran once again for his great talk and coming today. Great.